Okay, everybody, welcome to where we are, which is 224 Lab. What are we doing? Digestion, the digestive system. So in this lab, as you just heard in the introduction, we have some physiology, I think some fairly straightforward chemical digestion of the big three, lipids, proteins, and carbohydrates, then some anatomy, and some histology. Hopefully the anatomy and the histology will be fairly straightforward for most of you who have probably already heard some of this in your lecture, we hope. The physiology also should probably make some sense to most of you, but because of the differences in timing for the various lecture sections, let me explain a little bit. So what we're going to do as a trial here, you'll actually watch me do it right over there in the lab, is digest lipids, digest proteins, digest carbohydrates, using a few hydrolytic or hydrolase, little lecture for you, enzymes to digest these large polymers, at least in part. Lipids. To digest lipids is fairly straightforward. Almost any lipase enzyme will do the job if the lipids have been emulsified first. Remember that. Lipid digestion, chemically speaking, is pretty easy once you emulsify the fats. We will use pancreatic lipase as our enzyme, and we will digest lipids or triglycerides into their component monomers, monoglycerides or glycerol if you prefer, and free fatty acids. Now I'm going to emphasize this a little bit. It'll make more sense later, I hope. Free fatty what? Acids. We liberate fatty acids by hydrolyzing or breaking apart the chemical bonds in a standard lipid or triglyceride, releasing monoglycerides and free fatty acids. But again, we have to emulsify those lipids first. Proteins, very large, many monomers, big polymers. All we will do with protein digestion, excuse me, I tripped on a cord there. All we will do with protein digestion is digest large proteins into smaller proteins. This is what pepsin does for us. The same exact digestive enzyme or protease enzyme that we have in our stomach. Thank you, gastric pits. So we will watch how pepsin digests proteins into smaller proteins. It does not take us down to the monomers or the amino acid range by any stretch of the imagination, but it will do some chemical digestion for us. And last but not least, our carbohydrates or polysaccharides. In lab, we will use the fairly typical starch molecule, and we will use salivary amylase that's going to come right out of here, my own saliva, to digest polysaccharides into simple sugars, meaning di and trisaccharides. For us, we're really shooting for maltose, you can see there in your lab manual. Not all the way down to monosaccharides. That doesn't happen with the amylase enzymes. So salivary amylase digesting a polysaccharide starch into simple sugars. You may not say sugars. You must say simple sugars, indicating smaller di and trisaccharide units. Since I'm the guy writing your lab quiz and the guy writing your practical, you cannot say that salivary amylase digests a polysaccharide into sugar. Has to be simple sugars, or you could actually name the one we're shooting for, which is maltose, a disaccharide. So think about this. Maybe you even want to rewind to this point in the video, the online lab, after you see the physiological experiment happen, to understand what's going on here and the interpretation of the results. So I will show you each of the reagents we use. I will show you the indicators, Lugol's, Benedict solution, and how to interpret those as we perform the physiology. And the beautiful part for you, we can condense the time. So whereas normally you would be extremely hard-pressed to finish this in a 3-hour lab, here, not only can you fast forward, but I can cut and edit for time, so you don't have to sit around and wait for these things to incubate. I will do the waiting for you. So, let's not wait anymore, and let's get over there and start digesting. Hey everybody, is Professor Allner having too much fun in lab here by himself? Maybe. But let's get serious for a minute and talk about digestive anatomy. As you can see, I have a lot of various models for these digestive organs. Complete digestive systems on a plaque, my torsos, isolated liver and pancreas, and even teeth. Different models that I will show you for some of these structures. Now remember the rule, it still holds for the digestive lab. Any model that I show you here in the instructional video or in the practice video is fair game for your quiz or your practical. I'm going to say that again. Any model you see in this instructional video or in the practice video, and they'll be the same pretty much, is fair game for your quiz or your practical. So make sure you're going through both of them. You may want to watch that practice video a number of times for this digestive anatomy. Now this lab, unlike some others, has a bunch of things that you probably already just know. I bet you could point out the tongue. I bet you could point out the stomach, the liver on a lot of my models. But you do need to focus your attention on those anatomical structures that are new to you, that you haven't heard of before. So as I go through these, be following along on your list, and if there's one that you feel requires specific study attention on your part, circle it, highlight it in whatever color is appropriate for you. That way, you know to focus on that one when you watch the practice video. So get the most out of the online lab, or at least as much as you can out of an online lab, by being able to fast forward, freeze me, replay me, to focus on those anatomical structures you don't already know. So let's get started. Let's begin our anatomical study of the digestive system at the top left of page 6 in your lab manual, with some of the things you find in the head, in the oral cavity, and accessory to the oral cavity. Now you see these two torsos that I have facing each other right here to show you these two structures. The first of our salivary glands, these are the parotid salivary glands. The parotid salivary glands found in your cheek. Now, I show you this on these two models because every model shows a parotid gland. I tend to like the ones where it's colored quite a bit differently than the other structures, namely the muscles. They aren't all that way, believe it or not. So everybody has a parotid salivary gland out here. But not everybody has the other two, the submandibular and sublingual. So let's move this torso out of the way and do an about face with this one. Now, if you look closely, and I can zoom in for us a little bit, in this view, on this particular model, I can easily see the other two salivary glands. The submandibular right here, and the sublingual, meaning under the tongue, right there. So 3 salivary glands we must know. The parotid gland, lateral here on the cheek, every model has one. The other two, a little more rare to find good ones, but I see them right here. So look closely. Submandibular and sublingual. In fact, I can even see these little ducts going up under the tongue for my sublingual salivary gland. One more time. Submandibular, it hooks, curves under your mandible, and sublingual directly under the tongue. So 3, count them, 3 salivary glands. We also have in this oral cavity, I think, some fairly easy structures that I could ask you on just about any model. Lower lip, upper lip. Are you actually going to miss that one on a quiz or practical if that randomly comes up? I hope not. Lower lip, upper lip. The next structure you see listed, I mentioned back in lab number 5 with the respiratory system. That is the vestibule. The area between your teeth and your lips. This is the vestibule right here. I can see it right there on this model. I will zoom in a little bit for you. This is the vestibule right here between the teeth and the lips. It is the place where you would stick in a chaw. For those of you who might know what a chaw is, which is a wad of chewing tobacco, provided you don't care about stinky breath, oral cancer, bad teeth, and so on. That's your vestibule. The sound I just made, believe it or not, is a squirrel distress call. I'm from the Midwest. We do things like that there. By putting air into my vestibule, I can make that sound. That's a sound that a squirrel makes when alarmed. But it requires putting some air in the vestibule. I can also see the tongue quite nicely on this model. The hard and soft palate are also visible here, but probably a little better seen on our old head and shoulders model. So I'll grab one of those. So hopefully you remember this thing right here. So I can see the entire oral cavity, a great big tongue right there, my vestibule here and here, and I can also see the hard palate, we already know, and soft palate ending in the uvula. Don't want to bore you with this too much because you've already been quizzed over this, haven't you? Hard palate, soft palate, uvula, and tongue. Notice I do not see any kind of good salivary glands here except for the parotid, which everybody has one of those. What we're looking at here is a beautiful plaque model, just thin plastic. It's one of the older models in here, but boy, is it good for a tooth. In my humble opinion, and I don't think anybody here will contradict me. Do I have any naysayers? Hearing none, I move on. I think this is one of the best teeth models we have. So let's talk about the parts of a tooth. So we're about halfway down the left-hand side of your page here, and let's talk about some of these structures. Dental hygiene wannabes, this is all for you. The first structure you see listed is the crown of the tooth, which is the portion of the tooth that is above the gum line. So you can see the gum line ending right here, and everything above that between my finger and my thumb right now, this is the crown of the tooth. The crown is composed primarily of tooth enamel. You know, the hard stuff. Hardest biological substance produced by a human. Now, biological substance, notice I said. Now, here we have this enamel, which is sort of a, I don't know, cross between gray and beige color here. And then I see this yellowish material here. This is all dentin, which makes up the majority of the tooth, is dentin. Both of these substances, dentin and enamel, are very close relatives of hydroxyapatite. You might remember that one, CA10PO46OH2, from good old 223 way back when. These are similar to bone. Dentin, slightly harder than bone. It's probably about 70% or so hydroxyapatite, which makes it a little harder than bone, which is about 65% hydroxyapatite. Enamel, which is about 95 or more percent hydroxyapatite and much larger, more stable crystals. Enamel is a very, very hard crystallized version of hydroxyapatite. Enamel is considered to be non-cellular. Dentin, depending on who you talk to, is either cellular or non-cellular, because those odontoblasts, remember from your lecture, have finger-like projections that go in there. For lab, you simply have to be able to recite some names. Enamel is the material here. Dentin is this yellow material. The crown is this portion of the tooth that's above the gum. This is called enamel, which makes up most of the crown. So the question you would have, obviously, as a student in an online quiz, is how do you ask the questions? Well, you have to listen to my wording. If I have this indicated and I ask you for the material, what substance is this? It's enamel. If I have this section of the tooth indicated, easy to do in an image, that's called the crown. Dentin, enamel, crown. This pinkish material you see here is the gingiva, the gum. So this is the gum or the gingiva. And the section of the tooth, watch my fingers go all the way across, everyone. The section of the tooth that's where the gum is located, right here, is called the neck of the tooth. So we have the crown here, not the Netflix show, crown of a tooth. The neck right here, and the root of the tooth below that. So crown, neck, root are the three regions. Enamel, dentin, pulp cavity right here, where you see these blood vessels, some nerves, some connective tissue in there, and then follow my pointer down, extending down, just look where these blood vessels run. This is the root canal. It's the name of a medical procedure. Also, somebody drills in there and bores it out, called a procedure called a root canal, but the actual anatomical structure, this canal within the root of the tooth, is the root canal. So just one more time, the structures: crown, neck, root. Enamel, dentin, pulp cavity, root canal, gingiva is the gum. So with the regions, I will mark it so you can see crown, neck, root. But remember, what question I ask you will dictate the answer you select. Material is dentin. Material is enamel. So remember that as you look at the tooth. I have other models of teeth as well, like this thing right here. But you'll notice this one does not show me any gum. So it'd be kind of tough to tell where the neck is. If I open it up, I can see very nicely enamel dentin, the pulp cavity, and the root canal. But this one, an isolated tooth, don't have any gum line, kind of hard to tell exactly where the neck would be. I would need a model like this one that shows me the gingiva, the gum, in order to see that. So very nice tooth here, and a very nice tooth here, but limited in what I could ask you. On this model, we once again see some of those oral cavity structures. I think fairly easy for us. Lower lip, upper lip. This again is that vestibule. The tongue, hard palate, soft palate, and uvula. Those aren't too bad and fairly easy for us to see. Just about every model has those pretty well shown. What not every model has very nicely is one of these, the esophagus. I can see right here that muscular tube that is behind or posterior to your trachea. This model shows one quite nicely right here. So here's the esophagus. And then if I pull off this front part of the stomach and adjust my camera a little bit, I can see at the bottom of this esophagus the cardiac sphincter. Now you notice in your lab manual it's called the lower esophageal sphincter, which means of course I have one up above too, which is actually some constrictors of the pharynx. But most people call this the cardiac sphincter. So that's what we will call it here in lab. This is the cardiac sphincter right here, the sphincter muscle that's at the entrance into the stomach. You probably picked this up in your lectures, that these digestive organs we have at the front end a sphincter muscle and the back end a sphincter muscle. So here in the stomach, I have the cardiac sphincter, which guards the entrance into the stomach. And over here, right here where my pointer is the pyloric sphincter, which guards the exit from the stomach. Cardiac sphincter, pyloric sphincter, here in the stomach. And you can see in this stomach's wall all these folds, these sort of irregular accordion-like folds. If this was a heart, you might call these trabeculae carnii. Yes, I'm taking you in the Wayback Machine there. But these are the rugae, these folds in the wall of the stomach that allow it to expand and contract. So I can see right here the cardiac sphincter. I can see rugae in the wall of the stomach. I can see the, whoops, let me zoom in a little more, the pyloric sphincter right here. And the stomach has different regions that we like to describe in this organ. It's not the largest of your digestive organs. Certainly that distinction goes to the small intestine, believe it or not. But the stomach does have regions that we like to name. So follow along with me. Above the horizontal line formed by my probe is the fundus, right here, the dome-shaped top of an organ. If I put the face back on this stomach, like that, the fundus is the dome-shaped top of an organ. Your stomach has one, your urinary bladder has one, your uterus has one. So any dome-shaped top on an organ, that's called a fundus. This area right here, the entrance into the stomach where my finger is, this is the cardiac region, or sometimes called the cardia. Right here where my finger's waving around. So above my finger, the top, the dome, fundus. just a slightly thrown back view here. Above my finger, right here, the fundus of the organ. Right here, this small region just inside the cardiac sphincter is the cardia or cardiac region. Down here, just before the pyloric sphincter, is the pyloric antrum of the stomach. And everything in between is called the body. We also have, and I think these are the last two structures listed in the stomach, two curvatures of this organ. Which one do you suppose is the greater curvature? Well, obviously it's this one, the much larger one. So this is the greater curvature of the stomach. This is the lesser curvature of the stomach. Now, what I want you to be in mind of, or be mindful for on a quiz or a practical, I can show you these regions. I can animate this region right here and say, name this region, that's the fundus. Name this region. I could highlight this whole section here. The region is the body. Just right here, what's this region? The cardia. What's this region? The pyloric antrum. What is this structure? Not region. This is the greater curvature of the stomach. What's this structure? The lesser curvature of the stomach. This structure right here, the doorway in, cardiac sphincter. This structure right here, the sphincter on the way out, pyloric sphincter. These wrinkles inside, rugae. So regions, fundus, cardia, body, pyloric antrum. Structures, cardiac sphincter, pyloric sphincter, greater curvature, lesser curvature, rugae. And what is this region right here? Notice I said the word region, the body of the stomach. All at the bottom of what organ? Right here, the esophagus. Let's just take a quick look at these same exact things on a different yet very similar model. What's this? The esophagus. What's this organ right here? Stomach. What region of the stomach is this? The fundus. If I take the face off this stomach and zoom in a little bit, start naming some of these structures and regions in your head as I adjust the focus for you. What is this structure right here? Cardiac sphincter. What is this structure right here? Pyloric sphincter. This region, pyloric antrum. This region, the cardia. This region, the body. This structure, greater curvature. This structure, lesser curvature. And these structures, rugae. Hopefully learning the names of some of these anatomical parts will help you understand the digestive process for your lecture test and or your practical. So that takes us through the esophagus and the stomach. Next up, let's talk about some intestines, small and large. Now, I focused in on the abdominal or abdominal-pelvic cavity of this particular model, and I'm going to remove the liver, because we're not talking about that yet, so we can see this stomach again. And just like the other stomachs we've seen, has the same parts, fundus, greater curvature, lesser curvature, they all look the same. Cardiac sphincter, pyloric sphincter, cardia, body, pyloric antrum, and I can see some beautiful rugae in there. But if we were following food through our digestive tract or alimentary canal, where does it go after the stomach? It goes into this thing, which is the small intestine. Not this one, the dark gray one, that's the large intestine. The small intestine is first, and it's actually much bigger. There's nothing small about your small intestine, as you probably learned in lecture. The small intestine has 3 sections. Again, I assume you heard about these in your lecture, or you will. The first section is called the duodenum. It's only about the first 10 inches to foot. This right on the other side of this pyloric sphincter. Many of these models show it, but first you have to remove the stomach. You have to remove part of the large intestine. And then I can see right here the duodenum. Now there are a couple things I want to point out to you. Frames of reference, I guess I would say, anatomical landmarks that I think you should be aware of if some nasty person, namely myself, asks you to identify a duodenum on a model like this one. So there's no stomach here. I can't see a pyloric sphincter. It's pretty hard to orient where I am unless I see this pancreas right here. And notice it has a duct that dumps into the duodenum. The pancreas, as well as the liver, are accessory digestive organs to the duodenum. So if I see this sort of lightly colored pancreatic duct right here, and it's dumping into the small intestine right here, I don't care what else I see, I know this is the duodenum, the first section of the small intestine. The second section of the small intestine is the jejunum, which is all the way out here. This is all this stuff is jejunum. Very tough to tell one particular area from another. These loops of intestine held in place by the mesenteries. And the third and last section of the small intestine, look here in the person's lower right quadrant, right here, is the ileum. I-L-E, not I-L-I. I-L-I is the bone. I-L-E is this section of small intestine. So the last section of small intestine, look down here, be right down here at the right, this is where we'll see a few other structures, is the ileum. Let's look at it in another model. So just moving right next door to this big old-fashioned torso, I take off the liver here. I can see the stomach. Nice. Oh, look, it even shows me layers of musculature in the stomach's wall. Rugae. Greater curvature, lesser curvature. Not a great stomach, but it's all right. If, however, I look closely, see the pancreas and the pancreatic duct dumping into what? The duodenum. Now I can remove part of this large intestine, but you see it's hard to orient here. Look for that pancreas, look for the pancreatic duct dumping into the duodenum. This would have to be the duodenum. This first loop or turn of the duodenum is where your pancreas lives. Remember that. Look for the pancreas as a way to orient the duodenum in one of these models. Jejunum and ileum down here, look in the lower right. You'll see it a little better, I think, on two of my other models. So here we are back to the plaque model, which is an isolated or just the digestive system. This one, it's a little easier to pick out the three regions of the small intestine. So again, there's the stomach. Pyloric sphincter would be right in here. And what's this first section going into the first turn of the small intestine? This is the duodenum. I can see the pancreatic and common bile ducts coming down to it. So look for that pancreas living in that first curve of the duodenum. Duodenum, pancreas, see the white pancreatic duct. Whoops, nice and easy to see on this particular model right here. Then going down, I have jejunum all out here, and notice right here where my finger is, the end of the small intestine and start of the large. See where they're connected right here. This is the ileum, the last section. In your lectures, you will memorize who's 12 feet and who's 10 feet and who's 1 foot, all that stuff. But here we have, or 8 feet, excuse me, we have the ileum right here. So these sections are 1 foot, 8 feet, 12 feet long as a relaxed organ. It's hard to tell in here in the middle. So anything in here in the middle is jejunum. Up here by the stomach is duodenum. Down here by the large intestine is ileum. Same thing in one more model. So again, I can see the stomach. If I remove this piece of large intestine, notice the pancreas in the first curve. This is the duodenum, pancreatic duct. Down here in their lower right quadrant, I can see the ileum connecting to the large intestine. Everything out here is jejunum. These sections of the small intestine are fairly long, 1 foot, 8 feet, 12 feet in length if they were relaxed. It's very tough to tell out here where the jejunum starts and the ileum begins. So remember, for your quiz or practical, right here by the stomach, this first curve, duodenum. Down here by the start of the large intestine, ileum with an E, and anything in the middle, jejunum. Now I would like you to focus your attention right down here on the ileum. And in an online lab, I can essentially force you to focus your attention down here by simply zooming in, like so. So what we can see is the ileum, the very last section of the small intestine, as it enters into the large intestine right here. And that takes us, of course, to the large intestine. And you say, Dwayne, I'm following along in my lab manual, and it appears that you just skipped right over the pancreas. Oh, no, I did not. We're going to come back to that one, so don't worry about it. But right down here, at the end of the ileum, an entrance into the large intestine, I see this structure right here, which is another one of those sphincter muscles. This one, where my finger is right here, is the ileocecal valve. The sphincter separating the ileum and the cecum. Ileocecal valve. Following that pattern we see with digestive organs, way up north in this model, I had the pyloric sphincter, which was the entrance into the duodenum. Here I have the ileocecal valve, which is the exit from the ileum. And then we're in the large intestine. This little dead end pouch that we see right here is called the cecum. It has hanging off of it your appendix. So this is the appendix or vermiform appendix right here. This is a little thing in the lower right-hand quadrant that sometimes becomes inflamed, may rupture, and so on. You talk about that in your lecture, I'm sure. This structure right here is the appendix. This structure right here is the cecum. Cecum is a dead-end pouch on the digestive system, and this certainly qualifies. Moving this way, we next enter the ascending colon, going up on the right side. The transverse colon going across. The descending colon going down the left side. Then the sigmoid colon down here, where it takes essentially an S-shaped turn. I'll find us a better one. Followed by the rectum right here and the anal canal right there. So through the structures on this model, let's take a look. We have, to start out the large intestine, the ileocecal valve. The cecum and appendix, ascending, transverse, descending colon. Sigmoid is pretty weak here. Rectum, anal canal. I can also see very nicely on this model the two other structures. People confuse these once in a while. I'm not sure why, so let's be careful. Do you notice these pouches that you see here, this pouch-like appearance that the large intestine develops? These pouches are called haustra. So that's what these things are. Each one of these individual pouches is called a haustra. And this, notice the different color here on this model, this band of longitudinal smooth muscle fibers that makes a line right here on the large intestine, this is the teniae coli, tensors of the colon. As that material, changing from chyme to feces as it moves through here, becomes drier and drier, gets harder and harder to move it. So we create force with these teniae coli, but that makes the organ look rather puckered up. Let's go through these structures on the next model over, and we're going to find a better sigmoid colon as we go. So again, structures of the large intestine. Here was the ileum, ileocecal valve right there. Notice the great similarity in my models. Cecum, appendix, ascending, transverse, descending colons. And notice this one where it curves right here. This is the sigmoid colon. The rectum, between my fingers right here. and the anal canal right there, a little easier to see. Rectum, anal canal. I can also see on this large intestine the Tenier coli right there and all of these haustra. Let's look at one more. So large intestine right here, large intestine. If I remove this small intestine, get that out of the way a little bit, I can see what this model shows me. It's got kind of a nifty little door that flips up right there to show me this ileocecal valve, this cecum, and notice the appendix right down here. Ascending, transverse, descending colon. And right here, notice the curvature, the S-shaped curve right down here. This is the sigmoid colon. This model does not have a particularly good rectum or anal canal, but the sigmoid curves like an S right here is pretty good on this one. Ascending, transverse, descending colon. Notice all the haustra, the Tenier coli. Make sure you're paying attention to that in the practice video. Before we leave this large intestine, let's talk about these two flexures that we see, which is a perfect segue or transition right into the pancreas and the liver. So it's not by accident that I'm going through these in the order that I am. So you notice the liver here, name the organ liver, easy, name the stomach, or excuse me, name the organ stomach, name the organ large intestine. But notice where the liver is. Notice how the ascending colon comes up and it makes a basically 90-degree turn right here below the large lobe of the liver. This is the hepatic flexure, this corner. So I'd have to indicate the corner with dots. This is the hepatic flexure. So it would have to be both part of the ascending and part of the transverse colon. Hepatic flexure by the liver. Now, if I pop the liver out and the stomach off, notice this thing from way back in 223, this is the spleen. This corner right here is the splenic flexure. So I would have to use dots to indicate this corner right by the spleen, including part of the descending and part of the transverse colon, the splenic or splenic flexure. Move over here to this wall plaque model, which is very nice, but you notice no liver. I do, however, see a spleen. Splenic flexure, hepatic flexure is the other side. So even without the liver, I can still tell the hepatic flexure is on the right side of my person when they face me in anatomical position. Splenic flexure is on the left side. If I come back to my torso model, now this brings us to the pancreas. And you will notice, if we again think all the way back to 223, you know, back in those days when maybe lab was in person for you and you had to learn the names of organs. So the name of this organ right here is the pancreas. And if you had 223 with me, I know I told you this, I tell everybody this, the pancreas is posterior to and inferior to the stomach. I say that in 223 to force 223 students to learn their directional terms a little bit. So posterior and inferior to the stomach, the pancreas. Looks sort of like a little mass of brownish colored scrambled eggs to me. and I can see running down the middle of it the pancreatic duct. Let's look at my other two nice models here. If I remove the face of the stomach, didn't help my view a lot, I can see the pancreas and pancreatic duct right there. Let's go to this model also, and I can see the pancreas, and in white, pancreatic duct. Now, there's another structure here called the major duodenal papilla. The major duodenal papilla. This is the little bulge in the duodenum where the pancreatic duct empties. Not many models show a really good one of these, but this one certainly does. So I want you to notice what I have right here. Pancreatic duct. And I want you to notice right where it ends, right here where the tip of my probe is. This is the major duodenal papilla, thus named because it has the common bile duct and the pancreatic duct fusing right here, where the liver and pancreas are accessory to the duodenum, right here at this spot. So right here, where the tip of my probe is the major duodenal papilla. This is a pretty decent one on this model right here. And this green duct, remember the color green, will lead us into our study of the liver. What we see here in this abdominal pelvic cavity is sort of your standard view of a basic human liver. This dark, brick reddish kind of organ, I always refer to the liver as one of the three blood organs we have, so heavily infused with blood at any given time, you could bleed out pretty easily if you lacerate this thing. The liver is one, the spleen back here is one, and your two kidneys. Three very heavily infused organs. Their colors even show us that in a living person. If we look at the liver from this view, in place, in its normal anatomical location, I can easily see the much larger right lobe and the smaller left lobe, separated by what you see here as the two white lines, the falciform ligament. So again, just going through the anatomy here. Right lobe is the big one, left is little. Falciform ligament. I can see a little gallbladder poking out there too. Look at this model. Same thing. Right lobe, left lobe, falciform ligament. I don't think that's too tough for us, but when we start getting into all the duct work and these other structures in the liver, it can be pretty tough. That's why I have this massive super, I call it, liver model. So let's take a look at that one. So let's look at this liver model right here. I can easily see the right and left lobes, and no, a living person's liver is not supposed to have a big rod sticking through it. That's just so I can put it on a stand. The benefit of this liver model is our ability to see the underside of it. So in a living person, this liver would sit like so, And what we've done is removed it and turned it upside down so that you and I can see these structures. So now, imagine this person is facing the far wall, facing away from us. This is the right lobe, it's much bigger. This is the left lobe, much smaller. I can see the falciform ligament right there. And this view right here for these structures, this is the one I will show you on your quiz and your practical. This look right here is the one you're going to see. This is the look I will use in the practice video as well for some of these other liver structures. So first off, I want you to notice the big green thing. This is the gallbladder, storing that bile. Right? The target of CCK. A little lecture for you, just because it's fun. So that's the gallbladder. And notice, coming out of the gallbladder right here is the cystic duct. Cystic, referencing gallbladder, just like hepatic references liver. So the gallbladder is this big green bag. This tube coming out of it is the cystic duct. Part of, notice all the green, I told you to remember green, hepatic ductwork carrying bile, those bile salts, to go down to the duodenum and help us emulsify those fats. So if I look at some of the ductwork here, notice all the green over here in the larger right side. Not the cystic duct, but all these green ones. These are right, in the right lobe, right hepatic ducts. Now, some of my colleagues will gripe at me when they see this video and they'll say, oh, you can't name it like it's all one. I just did. So, all these ducts over here in the right lobe, right hepatic ducts, which continue to merge into bigger and bigger ones. Over here on the left side, I see what? Left hepatic ducts. These are the green ones, everybody. And where everything comes together, see this right here? See this left one shooting over here? See all these rights shooting over here? Where everybody comes together right there, it's cut on this model, but where everybody merges, this is the common bile duct. This is the thing that's going to shoot down to the duodenum. If I just slide a little bit over here, zoom in quite a bit, see this green right there? That's the common bile duct going down to the duodenum. Watch for that in the practice videos. So if we come back to this thing, just one more time for review. Gallbladder, cystic duct, right hepatic ducts, left hepatic ducts, common bile duct. And as long as we're here, we're going to name a couple of our veins of digestion. Look down at the list of veins of digestion. We have 3 of them that can best be seen on this model right here. Those 3 are the cystic vein, the hepatic portal vein, and hepatic veins. The only artery that we really see here is hepatic arteries. So, follow along with me here. Notice where my probe goes. To this large blood vessel right here, which is what I always call a goofy color. It's not red like our traditional arteries. It's not blue like our traditional veins. What color would you call this? I don't know. Maybe some kind of burgundy? Maybe a shade of violet? Maybe we could put our mask on and take a trip to a paint store and get somebody to tell us what this is actually called. But it's not red, it's not blue. This blood vessel right here, best seen on this model, this view, this is the hepatic portal vein. You talked about it, I bet, in your lecture. This is the vein carrying blood from the digestive organs into the underside of the liver for detoxification. Your liver doesn't filter anything. Professor Allner just said so. It does detoxify, however. So this thing is the hepatic portal vein. Notice the same colored blood vessel running across the top of my gallbladder. This is the cystic vein. Cystic for gallbladder. Everybody with me? Remember that. Same color, this weird purplish hue. Cystic vein, hepatic portal vein. Now, the other vein that we have here on this model, I like to use this top view to see. This blood vessel is your inferior vena cava. And notice this one going into it right here, that's one of your hepatic veins. That's a hepatic vein. I can see lots of blue ones down here, hepatic veins, but I always like, and remember who's making your quiz, this view right here, vena cava, inferior vena cava, and hepatic vein. We have one artery that we can see here, and that's the hepatic artery. Right there, and a little branch shooting off of it, the cystic artery right there, so I guess that makes two. Hepatic artery would be all these red ones. This happens to be the common hepatic artery, but we just have hepatic artery. So red hepatic artery, red cystic artery, purple cystic vein, purple hepatic portal vein, and again, this is what I like, hepatic vein right there. The liver. Very interesting organ. We only have a couple other digestive blood vessels to know here, and there aren't that many models that show them well. Those blood vessels, namely, that we have left, are the celiac trunk and the superior and inferior mesenteric arteries and veins. The celiac trunk I can show you on this model. So look, there goes his liver, hepatectomy, gastrectomy by stomach, colon resection, take off this intestine, and what I'm going to do is zoom in about as hard as I can right here to start with. Right here. So let's remove all this mesentery sheets right here. So notice the diaphragm on this model, right where I'm running my fingers, separating the thoracic aorta, which would be above, abdominal aorta below. I want you to notice something. Shooting right off the aorta, right here, right under the diaphragm, is a large artery that has three big branches coming out of it. This thing, right here where my finger is, this is called the celiac trunk. It has a splenic branch, it has a gastric branch, has hepatic branch. This whoops, right here, that's the celiac trunk. You will always know it because it is just below the diaphragm. The celiac trunk, right there. The other blood vessels of digestion, let me give you a nightmare flashback. Over here, oh no, look, it's Wireman. Wireman is back. Now, I just want you to notice something on the Wireman model from this sort of side view right here. Do you see an artery and a vein? Notice there's red there too. Right here, one above, one below. These are the superior and inferior mesenteric artery and vein. The Wireman tends to get bent up. You remember I'm not that fond of him, but I can see those very same blood vessels quite nicely right here. Let's tuck this back in. So here's what I want you to notice. What you see right here is a sheet of mesentery. You learn about that in your lecture. And I can see an artery-vein pair right here above and then right here below. Here are the superior mesenteric artery, superior mesenteric vein. Here are the inferiors that are lower. Superior mesenteric vein, superior mesenteric artery, right there. Inferior mesenteric vein, inferior mesenteric artery, right there on this model. So notice this one above, this pair above, this pair below. Superior mesenteric artery and vein, red and blue, traditional colors. Inferior mesenteric artery and vein down here, all these. Hopefully you caught that, but we'll go through it a number of times in the practice video. Here's just one more view of the same blood vessels. Notice the superior artery and vein, notice the inferior artery and vein. In this model, do you see how the veins are not your traditional dark blue? That's just because these merge together, forming that hepatic portal vein going up into the bottom of our liver. Remember, that thing's always some goofy non-red, non-blue color. So superior mesenteric artery, superior mesenteric vein, inferior mesenteric artery, inferior mesenteric vein. Notice how far down these go. Okay, now let's start talking about the last section of our lab, which is histology. And you might recall me telling you that lab 6, this one that we're in right now, and lab 8, the last one of the semester, have the more tough histology, in my opinion. The first 2 slides we have are fairly easy. I hope I'll give you a couple different views. So here's the first slide. Taste buds. Now, all I can ask you is, 1, name this organ. This is a tongue. These structures you see right here are papillae, the little bumps you see on your tongue. Now, for this histology, I will do my best to create as fine an image for you as I can. And I'll show you some of these at a couple different magnifications. So what is this organ? This is a tongue. What are these structures right here? These are taste buds. Those chemosensitive structures, receptors we have on the tongue But notice they're located in these little valleys on the sides of my papillae, my tongue bumps. If I just move over a little bit on this slide, one direction or the other, I can see, again, exactly the same thing. A tongue or lingual papilla, and then these light-colored structures here, these are the taste buds. This is a perfectly good slide. Let's jump up in magnification a little bit. So what you see here, this is a taste bud, this is a taste bud, that is a taste bud. To me, these things look like, a rosebud or a tulip bud before they open. That's why they're called buds, taste buds. What I want you to look at is this slightly lower magnification we saw before, because this allows me to recognize the organ. I am looking at a tongue. These structures are the taste buds. So again, only two questions I can ask you from this slide. Name the organ, tongue. Name the structure, taste bud. Here's another perfectly decent view of the human tongue. Notice these papillae right here, and then on the sides, these taste buds, which aren't stained that great in this particular slide, but you can still see them. Let's jump up in magnification. Very nice view right here. So again, name the organ, tongue. What are these structures here and here? Taste buds. Here's our next histological slide. This one is salivary gland. And what can I ask you about this? Name it salivary gland. The thing that gives this slide away, and any other salivary gland that I show you, is this network of salivary ducts that you see. You don't have to name them, but I want you to look closely. I want you to look hard at this thing. And doesn't it sort of bear a resemblance to an aerial photograph or a satellite photo of a complicated river system, like say the Nile River Basin or the Amazon Basin, something like that? So whenever you see these, it looks like aerial view of all these rivers, salivary gland. And that's the only thing I can ask you from this slide. Name this organ, salivary gland. Okay, here's just more of the same. Again, I see these salivary ducts. Some of them cut in cross-section, most of them in this longitudinal section, which allows me to see the river-like look of this thing. This is a salivary gland. Here we have our third histology slide, the stomach. And remember what I told you in the brief histology intro, the stomach and the small intestine and the large intestine all share a common style of mucosa, but not common modifications. So this is a stomach. On scanning power, I can see a single one of the rugae, a rugus right here. Another one over here, these folds in the wall of the organ. I can even see a few gastric pits. Most of them are so narrow as to almost be unrecognizable as a pit. Let's look around a little bit on this slide. So here is one of the rugae. So if I put a dot right here, rugae. Notice the gastric pits here, a little bit of gastric mucus there. This layer, that is the epithelium that I can see right here, that is the mucosa layer. The mucosa layer right here. Innermost, the food would be right here. If I work my way down, I can see different shading here in this particular organ. Notice right under the mucosa is this lighter shaded area. This is the submucosa. The submucosa. This is all smooth muscle, the muscularis layer, and you can see right here, right where my meter stick is, there's a difference in the appearance. This is the serosa out here. So, a single rugae, I can see these tiny gastric pits. We will see some on higher magnification. Mucosa, submucosa, muscularis, serosa, the layers of the stomach's wall. Here is another stomach slide just to give us a slightly different view. And I can see a big rugus right here, one of the rugae, so you'd say rugae. And then all these narrow gastric pits shooting down into this epithelium or mucosa layer. Rugus, and these little, look almost like cracks shooting down here, these are gastric pits. Now, how would I know this is stomach versus, say, small intestine or something like that? Well, if I increase magnification, I think you can see what I told you about before. Look at all this simple columnar epithelium that I see everywhere. Notice, oh, I can see a goblet cell here, couple over there, but not much in the way of goblet cells. Look at this. Whole big section with no goblet cells in it at all. So yes, I have simple columnar epithelium, but very few goblet cells and these narrow gastric pits in the wall of the stomach. And let's take a look at the layers here. If I can get, there's a good spot right here. So notice again right here, a rugus, narrow gastric pits. This is all the mucosa right there. Under the mucosa, right here is the submucosa. And if I keep going down, look at this, people. A beautiful giant sheet of smooth muscle fibers. This is the muscularis layer of the stomach. And then at the far outside, where things start to look like they're loosening up a little bit, this is the serosa layer. Muscularis, muscularis, submucosa, mucosa, where those gastric pits are. The stomach. So I can ask you, name the organ, stomach. Name this structure, rugae. These little cracks, gastric pits, and then the four layers. Mucosa, submucosa, muscularis, and to the outside, serosa for the stomach. Here we have the next digestive organ. Remember, maybe from your lecture, those digestive organs are the ones that actually have food going through them. So this is your small intestine. These structures are the villi, finger-like projections of the small intestine. I can ask you those. So I can say, name this organ, small intestine. Name this specific structure, villi. I can also do the layers of the digestive wall, which is common to these digestive organs. So stomach, small intestine, large intestine have the same layers. Notice the darker staining mucosa, epithelial cells tend to take more stain. The lighter staining submucosa, then the muscularis, and the outer edge, the serosa. Muscularis, submucosa, mucosa. Now, I know what you will be thinking as you look at this slide and maybe some others of the small intestine is, how would I tell this from the stomach? Well, first, notice that these gaps between the villi are much wider than a person's gastric pits would be. But that may not be enough to tell you. Also, notice these sort of large clusters of cells here in the mucosa. These are called intestinal glands. You will see those in the small intestine. You don't see those in the stomach. And the third giveaway, down here in the submucosa, look at these clusters of cells that you see. These are the duodenal glands. So intestinal, small intestine glands, duodenal, duodenum, small intestine glands. You will see those in the wall of the small intestine. Name the organ, small intestine. Name these structures, villi. Name these structures, intestinal glands. And name these structures down here, again, notice in the submucosa, duodenal glands, the small intestine. Let's take a look at another small intestine slide. Here we have another slide of the small intestine that has stained a little darker. So obvious layers I can see: mucosa, submucosa, muscularis, and serosa. That's pretty easy. And I think if your eyes can't tell you now, I'll do a little magnification for you. You can see also characteristic in small intestine a higher frequency of goblet cells in the epithelium here than I would see with the stomach. If I go back to this standard magnification, I think you can still see those smattering occasional goblet cells. I can also see here some intestinal glands. You see those little white spots. What this slide does not show me very well, and I'm scrolling around, is much in the way of the duodenal glands. I can see a couple here, but not too many in this particular slide. So what's this? Small intestine, beautiful villi here, mucosa, submucosa, some intestinal glands, and a few duodenal glands, but we've probably moved out of the duodenum here. That's why there's so few here. But it's still small intestine. Pretty nice slide. Here is the last of our digestive organs, and of the three, stomach, small intestine, large intestine, the easiest, I think, to identify is this one, the large intestine or the colon. Why? Because there are goblet cells everywhere in this slide. I don't know if you can tell that from your point of view watching me online. Let me zoom in a little bit. Notice goblet cells all over the place on this particular slide. There are more goblet cells visible here than I can count. The large intestine has this. Here's another slide of a colon or large intestine, and I increase the magnification slightly. Look at all these goblet cells. As soon as you see that huge number of goblet cells, that's the colon, that's the large intestine. Couldn't be anything else. That's the only one with all those goblet cells. And of course, I can see the layers of the wall, mucosa, submucosa, muscularis, and serosa. Large intestine, and look everyone at all those goblet cells. What you're looking at here is a very, very nice, in my opinion, slide of a liver. Why is it so nice? Well, it shows me this structure right here, which is a liver lobule. You might say, hey Dwayne, how would I recognize this as opposed to a salivary gland? Rewind and look at the salivary gland. I see these structures here which remind me of those rivers. Well, what I want you to focus on here is this repeated pattern you see in a liver slide. These structures that are roughly hexagon shaped, always with a vein in the center called the central vein. This is a liver. I'll show you several. So notice this repeating sort of pattern with a hexagon and a central vein in it. Here's another one right there. It got cut off. Sorry about that. There's the first one we looked at. Here's another one. Here are a couple right here. So look at this, central vein, common for the liver. And all these little cracks, these are what are called liver or hepatic sinusoids. This is where that hepatic portal and arterial blood mixes and gurgles and gets detoxified from your lecture, not for lab. Lab, you just have to know this is a liver. This is a central vein, and these little cracks are sinusoids. And I'll show you another liver here in a minute so you can take a look at a different one. But livers are pretty easy once you learn to look for this pattern of these geometric blocks. So here's another pretty decent liver slide. Again, notice the block-like appearance, central vein, and sinusoids that are clearly or plainly visible in this slide. Here is yet another liver, just showing you, I hope these are easy to recognize, block-like appearance of these small lobules, central vein, little cracks are sinusoids, a liver. And that's all I get to ask you from this one. What we're looking at here is our final histology slide for lab number 6, which is the pancreas. We're going to see this slide here in lab 6. We're going to see it again in lab number 8. Now, let me tell you what to focus in on to identify this as a pancreas. I tend to like a slide like this one that stains pretty darkly. Notice how dark these cells have taken the stain. And then notice small little groupings of cells that are lighter. Those are structures we'll learn about in lab number 8. Those are the endocrine structures. Not important for us except to tell us this is a pancreas. So as I pan around this slide, it doesn't matter where I go, I still see these little groupings of lighter staining cells. They happen to be called pancreatic islets. We don't care in lab 6, but that tells me this is a pancreas. This is the slide we will see. We will see those lighter staining areas called pancreatic islets, pancreas. The only thing I can ask you besides name this organ is all the cells we see. The pancreas, as far as we're concerned in lab, has two populations of cells. The cells that make up the islets, and then everybody else, which are called the acini or acinar cells. So name the organ. As soon as I see these light islands, pancreas. Name the cells, pancreatic acini cells. Again, you will have to see these lighter staining masses to recognize it as the pancreas. Look for those. Don't get lulled into thinking, oh, this is a salivary gland or this is a liver. No, look for those. If you don't see those, it's not the pancreas. Acini cells, the organ is the pancreas. Here is another just randomly selected slide. This actually out of the tray that students would look at if they were actually here. But I see the lighter staining cells, pancreas. Name the organ, pancreas. Name the cells, pancreatic acini cells.